All right, welcome again to the National Pilot Webinar, the September call. We're going to go ahead and get started. Hopefully everybody had a great summer. Just as a reminder to everybody that these are our open discussion types of calls. However, if someone is presenting, please mute yourself. But if you have a question or a comment, feel free to unmute yourself and ask your question, or you can type it in the chat. And these calls are going to be recorded and they'll be posted on the NBSTRN YouTube channel, as well as our website. So for this month's meeting, we are going to get some updates from states that are conducting active or they have active pilots, or they've completed pilots and it's on their state panels. And we'll also get some other updates from conditions that have been added to other state panels or have been nominated to the RUSP. And then we'll have some updates from NBSTRN. And if you have update, feel free at the end, you can also unmute yourself and announce it. So I think we're going to start off with the New York program, and hopefully Michelle is on the call today to provide the update. Hi, Michelle.
Hey, everybody. I have a update for the Guardian study, and I have slides for that. So I know if you wanted to do it now or you wanted to do some
Sure.
other.
I'll stop sharing.
Okay.
Let me know. You should be able to share, but if you're not, I'll make you a co-host, so you can share.
I can. I think that's it. Okay, can you see that?
Yes, we are good to go.
All right. Do I have to stop there? Okay, good to go.
Now you're in presenter mode.
Did I do it the wrong way?
You were fine, and then you changed it.
All right.
There we go.
Perfect. Okay. Hi, everybody. Good afternoon. Good morning. Yeah, it's probably morning for some people still. I'm going to give an update on the Guardian study. This is actually, today is the 1-year anniversary since we began the screening for Guardian. I will say these slides were provided by Alvin Ziegler, who worked at Columbia, and he gave them to me to present to you today. So I just wanted to start off talking a little bit about what the study is. It is based out of Columbia University Medical Center. There's several hospitals, birthing hospitals, that are enrolling patients. It is set up so that patients are recruited or told about the study prenatally during the 3rd trimester when they come to one of the Columbia area clinics. They give consent postnatally, so this is a consented study. The newborn specimen that we receive DNA is extracted by our program and sent off for genomic sequencing at GeneDx. This is a collaboration between many entities, and at the end I have a slide acknowledging all of the people that are involved. And within 3 to 6 weeks, a clinical result is provided by GeneDx. It goes into the patient's Epic health record at Columbia, and also is amended to the newborn screening report. So a pediatrician can get the Guardian result when they look up the baby's newborn screen results. And then they stay with the newborn screening report in the future. And obviously the ultimate goal of all these types of studies is early treatment to improve overall health. So we have what we call two groups. Group 1 is offered to all participants, and that is a screen for 148 genes for 155 conditions. Most, if not all, of the newborn screening conditions are on here, and things that are actionable within the first 5 years of life, where there's a treatment available and confirmatory testing available. There's also another set of conditions we call Group 2. This is another 100 conditions, mostly neurologic type conditions, seizures, and patients, babies who have these diseases can benefit from early intervention, OT, clinical trials, and that sort of thing. And of interest, out of the first, out of our data so far, about 90% of the babies that are enrolled, their parents enroll them for both group 1 and group 2, which we found kind of surprising. We thought most people would go for group 1 only. So the idea, and this is set up in a collaboration with the newborn screening program, because we feel that this kind of a study is synergistic with traditional newborn screening. Both are better together, and there's definitely places for both of these. And so we did find that some of the conditions that are on the RUSP are difficult to sequence. So we have poor coverage in our panel for CYP21A2. On the converse, also from the newborn screening perspective, most of congenital hypothyroidism is not due to there's not a genetic cause for it. And so the traditional newborn screen is important. But understanding that the scope for potential conditions on newborn screening is wider when you look at genetics. So a couple of the ones that come to mind are things like Menkes and Wilson disease and some of those. We expect a higher positive predictive value with than traditional newborn screening, and that's because it is a Guardian is screening, but it's a little bit different. And we have some examples where actually we have false positives in both directions and false negatives in both directions. Sensitivity of genome sequencing is different for some of the conditions, as I kind of alluded to. And so again, we feel like these are synergistic. So looking at the enrollment right now, we have a total of 4,631 patients have been enrolled. Most of them are done by in-person enrollment. We start out with a pool of almost 10,000 eligible. Of that, we approach a subset, so about 64%. And then from there, we enrolled about 73% of the total eligible. We have results for the first 3,600. The others are all in process. So looking at the sequencing, we actually get very good results. We use anywhere between 4 to 6 3mm punches from the newborn screen card, and we get sufficient DNA to complete sequencing. We've only had 3 QNS, 6 have failed sequencing, and we've been able to successfully sequence close to the 3,600. This is just a quick summary of the results. We're running about a 4% positive rate. We have 4 babies who screened positive on Guardian for more than one condition. And the PPV is shown here. And this group 2 positive screens, we have 5 still pending, 1 pending for group 1. And you can see the true versus false positive, like I said, for both sequencing and newborn screening. So when you look at the true positives from group 1, the overwhelming majority of our positive cases are G6PD deficiency. One of those, 85, was detected by newborn screening. And that was because New York State has a law that sort of earmarks babies who are to be screened for G6PD. Another 4 for sickle cell disease, some hearing. We had a baby with achondroplasia who was found by Guardian, 2 babies with SCAD, again, found by Guardian and by our newborn screen. And the sickle cell, LC-04, was all detected by newborn as well. Going on, the next set of group 1s, we had a long QT syndrome, and this baby actually had a prolonged QT interval, and so is on being managed. We have 2 babies with Wilson disease, and one of them had elevated transaminases and is on treatment as well. We had an MCAD that's being called, quote, false negative, but interestingly, this baby had a borderline result in newborn screening, and then the second result came back within normal range. This baby also has an older sibling who actually was referred for newborn screening as a positive on MCAD, and was seen at the treatment center, the specialty care center, and was released from follow-up saying the baby did not have MCAD, or the child did not have MCAD. And so this is one of those instances where there's a variant that is detected on sequencing, but biochemically we're unsure whether or not the baby actually would ever show symptoms. And there's also another report in the literature with one of these variants in this child that shows a very similar result where the baby was not picked up by newborn. And then later on, an older sibling as well. We had another case of SCID, IL-2RG, and that also had normal TREX for us. I think it had about 400 TREX, which is normal for us. And we also found another report of a baby in the literature, a child who had the same exact variant, also diagnosed with IL-2RG SCID, and had some symptoms earlier on, but again, had a negative newborn screen, just like our child. We also have picked up a baby with a genetic form of hypothyroidism as well. And then as far as the false positives for guardian of interest here, CFTR, originally a set of variants that always occur in cysts for us were being called as positive by guardian sequencing. We know that we see these two variants all the time in cysts in newborn screening. And so this was reclassified by GeneDx as a haplotype. And so those are no longer going to be called positives by guardian. We also had some DUAX2 variants, and those again were negative on confirmatory diagnosis, and we made some changes to that gene and its interpretation as well. And then we had a false positive on sequencing for MPS 7. We also did, we have 3 more here, MPS 4A, another hypothyroid, and a Pendred syndrome. And again, these all were shown to be false positive by the sequencing call. The babies were follow-up tested and then released. When looking at the group 2 conditions, we had a couple Dravet that were false positives on the sequencing test. One of the false positives, in 2 of the cases, the variant was inherited from an asymptomatic parent. And so this just illustrates that even when you do sequencing, you can have false positives. There may be cases where there's de novo and you have to do additional family testing, or you find asymptomatic parents. And this is kind of reminiscent of also within newborn screening for ALD, where a lot of times we will have a baby who has a biochemical alteration, has a variant in the ABCD1 gene, and then ends up actually having an elder relative who's perfectly normal. And so it's similar challenges whether you're looking at biochemical or you're looking at sequencing data. And this just wraps up sort of the group 2 conditions and what we found. We have a Rett syndrome, a Rett-like syndrome, a Barrister-Winter syndrome, and a false positive charge. Again, mom had the variant and it was normal. And the Barrister-Winter also was paternally inherited, and the father was normal. So I don't want to take everyone's time, but in conclusion, we had a high enrollment rate. And like I said, about 90% of the enrollees agreed to do in both group 1 and group 2 conditions. We are illustrating that genome sequencing based newborn screening is somewhat feasible on a modest scale. Again, it's our 1-year anniversary, and we are at about 4,600 babies screened. That is less than a week worth of newborn screening in New York. Results are returned outside of the time critical region, obviously, for some of these conditions, which illustrates the fact that this is a synergistic model, but not a replacement for traditional newborn screening. And obviously that genome sequencing can diagnose treatable conditions, but also we find false positives in sequencing as well. So this kind of work requires a constant refinement of variant calling and interpretation and amendment to databases so that this information can be made available to those who want to do this type of screening in the future. The challenges are pretty well understood. Obviously, the scale up would be one thing that's pretty important that would be required for newborn screening. What we actually report, and as I said, the turnaround times. And so this sort of takes the village. We have a large group of people who are involved with this. We have the staff at GeneDx that we have weekly calls. New York State, we have Denise, myself, also Jennifer Bradley and Robert Sicko, who pull all the samples. Again, we're getting these consents come after or while traditional newborn screening is ongoing. And so someone has to go back in, re-pull specimens, and people in newborn screening know that's not trivial to be able to do. Also, staff at Illumina who play an important role in this project. And this is a picture of everyone involved. And thanks. And anyone has questions, I'm happy to try and answer them.
Thanks, Michelle. Very great presentation. We have some questions in the chat, and I see Dr. Priesty has her hand up. Dr. Priesty, you want to ask your question? Is also, you can also state your comment in the chat as well.
Yeah, no, just that great presentation, Michelle. And I had a question, maybe two questions, in addition to my comment about Dravet in the chat section. But I was curious about, you said something about G6PD is routinely screened or not screened in New York State. Can you say more about that? And is anyone looking at, this high, not necessarily high and unexpected, but it would be great to do some follow-up and determine, whether this screening approach for G6PD, makes a difference in terms of health outcomes for some of those babies.
Yeah, so in New York right now, we have a law it's in our statute for newborn screening that any baby who has high bilirubin, returns to the hospital for high bilirubin, has hemolytic anemia, is of the high risk population so far as the folks that are most frequently affected with G6PD. Any baby that checks one of those boxes is supposed to have G6PD screening, or actually the way the law is written, a diagnostic test done. And so of interest of the babies that we detected, there was only one that had qualified based on that assessment for G6PD. And that's why it was 1 out of 85. That law is in process of being looked at, in part due to that kind of a result that we got. And originally, I think the G6PD frequency was something like almost 8% of the Guardian population. Now it's back down. When we look at the 4,600 babies, it's down to about close to 2%. So somewhere in there is the reality. What was your other question?
I didn't say it yet. So my other question is related to, you mentioned like there was a haplotype in the CFTR gene that you've seen repeatedly. And I'm just wondering if there's any effort at maybe long read sequencing, or if there are other strategies to help eliminate those kind of false positives once you start to recognize that those are in cysts and not necessarily associated with a disease phenotype.
Yeah, it's R74W and D1270N, and they occur in cysts for us almost all the time. So when we get a result from newborn that has those variants in it, we always ask for parent phasing. And then we phase the parents, and out of all of our cases, they've always been together. And originally, because they are both pathogenic variants, they were being called as positive on the Guardian. And so when we looked at those results, we went back and they changed the output, the reporting for that. We are working on doing some long range. We are working on doing long range PCR to be able to confirm that without having to ask for parents, so that we would know ahead of time they were in cysts. And this is true for some other variants as well that we tend to see together. So that way we would be able to do that early on, and then we wouldn't report that baby as a positive for CF. In this case, we would be able to report it as a carrier if we found those two variants only. So that's a kind of a work in progress.
So you would do reflexive long read for those particular regions of the genome, essentially.
Right.
Okay, cool. Thank you.
Yeah. Let's see what else here.
Paul had a question about asking about other genes that are included in hearing screening.
Yeah, I believe there are others. I can put the list, a link to the list in the chat, and then you can kind of see the what's all on the group 1 versus group 2 panels. Yeah, and the penetrance is something we've discussed a lot about that. whether, you know, how, again, you're in a situation where you find something in a healthy child is just like newborn screening, and then you have to figure out if it's reality or not. So, we labeled it a false positive because right now there's no evidence that the baby would have that. The parents have the information and it's in the baby's medical record. Joe, let's see. Yeah, we're calling it a molecular false positive for now, just because there's, you know, we've done additional confirmatory testing and found it was negative, or and again, this goes back to the interpretation of the variant, right? So they use the criteria that everyone else uses in order to call variants, and we know from doing newborn for the diseases that we actually do sequencing for, we know that there are things that aren't necessarily pathogenic, but may be labeled as such by the labs that are doing the interpretation. And so that's kind of, maybe we can change the name, but right now that's kind of the best we have for it. And it is
I'm sorry.
Mhm.
I was just going to ask if you were reporting some of that information into any databases such as ClinGen, anything like that for false positives.
I'm not sure if that's being done in real time or not. I'd have to check, because again, all the interpretations are all being done by GeneDx, and I know that they do deposit in ClinVar. So and you know, our goal is also to work with the Eden Project, the CDC based newborn, and we can certainly add to that as well. And to Amy's question, perhaps it seemed like I took some slides out of Albin's presentation, but we also asked questions about enrollment and, you know, why they enrolled. And it was kind of interesting. Overwhelmingly, most of the people that enrolled said that they did so to help other babies. Right? I mean, there was actually fewer people that were interested in having the information available to them in the future, I believe, than where we're basically doing it sort of to help the cause. So I thought that was kind of interesting.
And Michelle, thank you. Can you remind us, are your sort of recruiters or enrollment folks just like we used in the Duchenne pilot? They're right there in the hospital?
Mm-hmm. Yes.
Because I think some of the other, you know, neonatal sequencing programs like this, like I think maybe in Massachusetts, BabySeq isn't the only project that they're getting asked to participate in, and they get asked to participate at different stages. So it just seems like you guys are really successful in no extra samples, doing it in the hospital. So I think just understand which models are leading to people signing up for things is really interesting. Thank you.
Yeah, it's true. Like if you, you know, I could imagine if, first of all, they're being educated early before the baby comes. And like you said, there's no extra sample that's necessary at that point. And this was when Wendy Chung was setting this up, she's the overall PI. When she set this up, a lot of work went in on the front end to make this acceptable through focus groups and community engagement and that sort of thing as well. And so that I think also helped with the familiarity of the program.
Great.
And if you're getting them bombarded, like you said, with a bunch of studies all at one time, it could make it a little overwhelming for parents, especially if it's being done right in the nurse, you know, right in postpartum.
Michelle, I had one question also as far as the timing. I know you commented on the timing, that it's not a replacement. So a lot of the results are after newborn screening occurs. Mm-hmm. Can you give me an idea of how much longer? And then I was also wondering for like G6PD, since you guys, there is a protocol, if you had any idea on how the timing works with those results as well.
So the Guardian results are usually done within 3 to 6 weeks after that's the turnaround time for the laboratory. Then we get those results back. We have to amend the baby's newborn screening report, and so that's sort of the time frame. So it's certainly not a 5 to 7 day of life deal, and that's kind of why I think it's important to keep reiterating that this should be that this should be a synergism, not a replacement necessarily, just due to the turnaround times on some of these, for some of these conditions. And what was your other question?
No, oh, it was, it kind of answered it, with because I'm assuming that your D6 PD protocol is still happening faster than 3 to 6 weeks.
Yeah, I mean, the idea with that is that, well, if the baby develops some of these things in the nursery, certainly, but if the baby goes home and comes back, I don't know how long bilirubin sets in, hyperbilirubinemia sets in pretty quickly after birth. And so it would be based on the turnaround time for the diagnostic labs. We're not doing that testing. It just happens to be in the newborn reg because it's a newborn thing. So the hospital's actually sending samples out to diagnostic labs. So the turnaround time for the G6PD is probably within the same time frame that it takes for this to come back, I would think, or probably a little sooner. Yeah, I don't really know. I would think it may still be sooner depending on when the child presents with the switch to, make that test occur.
Amy asking about long-term follow-up, but since this study is only a 1-year anniversary, can you just discuss if there's plans to continue to follow these and collect data on these infants long-term?
They're going to be seen as part of clinical care. I don't know
Okay.
that it's a formal long-term follow-up per se. Prenatal patients, so they're introducing this study in via the clinics that see families that are going to deliver in the Columbia system. So it's focused within New York City and these certain set of hospitals. I think there's educational materials. We're not as involved in the recruitment end, but there's materials that are presented in the clinics for within the Columbia system only. And 4% positive rate. It doesn't really impact newborn because the children are all being followed through Columbia at this point. Certainly the ones that we get, the dual hit on, we're already aware of by the time the guardian comes back. The pain points for our part, the pain points are going back into the specimens and pulling out the specimens, cross-checking the consent. We don't want to sequence a child whose parents didn't consent, and vice versa. Don't want to not sequence one who did. So there's a lot of effort that goes into cross-matching those. The DNA extraction we were doing was a pure gene. It was more manual. We have just validated a more automated DNA extraction and found that the results are sufficient, that GeneDx gets good coverage. So we're going to move to that, which will be a little bit easier. And then on the back end, Bob Sicko in our lab wrote a script so that when the results come back from GeneDx, now they automatically get put into the baby's file. Whereas prior to that, it was a manual matching. So that also was time consuming. So it's not necessarily the follow-up piece, but more that sort of front and back end work that we were doing.
Well, so it seems like we got through most of those questions. There was a lot of questions on that presentation, because it was a really good one. I won't go through the whole thing, the rest of the things you've been reporting with Michelle, but I was wondering if there was any updates on CCMV, your pilot for CCMV.
So that is getting ready to start. We're having a hospital information webinar tomorrow. We are waiting still for the brochures that have to go out. So everything else is in place, and that's what we're waiting for. We're hoping to begin prior to the end of September. And there's been a lot of we've had some issues just getting things approved and through, but we have been running the assay and it's performing quite well. And once we have our brochures out and we have the hospital webinar on Friday, we should be able to start fairly soon. So stay tuned.
And is there any, I know last week there were, or in June, they reported of the Screen Plus had found some Fabry or and Gaucher cases. Is there any new cases that were identified through that screening process or any updates?
I don't know. I
Okay,
think because we're doing Guardian, we didn't pull that data for this time.
that's fine.
Yeah, nothing really off the top of my head, Jennifer.
Oh, okay.
We had, we did have an interesting MPS2 case that was part of Screen Plus, and it's a female that is homozygous for a variant of unknown significance. We're waiting to see what the urine gags look like on that baby. The blood gags were actually slightly elevated, so it's an interesting case. Just gotta see what the follow-up shows.
Yeah, and
Thanks.
that baby
Okay.
is homozygous for the same variant as a baby girl, so it's kind of interesting.
Well, thank you for the updates. Any other questions for the New York team? Okay, I'm going to switch it up a little bit just because I know someone might be leaving the call early. Dean, I know you had an update as far as the MLD, and I was wondering if you wanted to share it with the group.
Yes, I am ready. Can you hear me?
Yep, we can hear you.
Excellent. Sorry, I'm out in my car, but I want
I think we
update
just lost
on
you.
metachromatic leukodystrophy and our work there. I'm Dean Sur. I'm president and co-founder of MLD Foundation. Back in 2005, we were introduced to a team in Milan that was developing a gene therapy. They started clinical trials in 2012, and 2 years ago got approval by the EMA for gene therapy for MLD. Orchard Therapeutics is a current owner of that therapy and has submitted a BLA to the FDA a little bit earlier this summer. It has not been accepted yet, but they're expecting that soon. So we're at that regulatory review stage. Newborn screening was developed by Professor Gelb, who I think is familiar to all of you. And we've been working with him, I don't know, better part of 10 plus years on the screen as well. And it looks really well. We're involved in Screen Plus and Dr. Wasserstein's program there and Dr. Orsini. And also there is a significant pilot that's been ongoing with over 100,000 babies screened in Germany for the past couple of years. In that screen, they have identified 3 MLD babies, 2 of which have been referred to therapy. The third one, I don't believe has actually been referred to therapy yet, but they're all positives. They wait about 6 months before the baby gets the gene therapy. So with all of that, babies identified, a screen that's been validated and at least approved in the EU and an FDA application in place, we are entering the later stages of RUSP nomination. So we're preparing that. There's a meeting in Europe in, I don't know, it's about 5 or 6 weeks with the folks over there, and we'll bring that data back, and that will be a key component of our application to the Secretary's Advisory Committee, and we hope to submit that in the first quarter or so of next year. So we're making great progress. We're excited to not just have a regulatory approval, because that doesn't mean anything if you can't diagnose and identify the patients, the babies in this case. We will be screening for at least the target right now. And again, the nomination is not done, but it's clear it will be late infantiles and early juveniles. There's discussion about late juveniles and whether they should be included as a primary identification or not. They will certainly be identified by the screen. And then the adult form of MLD likely will not be, well, it certainly won't be a primary condition for the screen, and there'll be discussion about whether that's a secondary or not. So with that, I'm happy to answer any questions. We will be at APHL with many of you in a couple of 3-4 weeks, and be happy to explain more there. We have 2 pilots we're working on in the US. I'm not going to give state names right at this point. It's a little bit early, but if you're interested in getting a head start on MLD newborn screening, we'll be there.
So Dean, I saw a post, and if anybody from Illinois is on this call too, feel free to also comment that MLD was added to the Illinois newborn screening panel, was approved by the governor. Do you have any information about that, or does anybody else on the call?
Hey, can you hear me?
Yes.
Oh yeah, this is a
Hi.
Vineet
Hi,
from
Vineet.
Illinois. Hey, yeah, so that was signed into law by the governor, starting of next year, January 1st, 2024. So as you can imagine, that's kind of thrown our priorities whack a little bit. So the plan is to start developing MLD. That's not something I'm personally involved in. It's another group lead, but the timetable is to hopefully aim Q2 2025 to kind of go live with that testing here after we're able to kind of get all the instrumentation, validation testing, and all that done, as well as working with PerkNumber or Revity as far, with our LIMS developer to get that all situated and built as well. So that's all I really have to say right now. A lot of it's still kind of in the planning stages and exploratory steps as far as trying to figuring out the timelines. But yes, that has been signed into law, so it's kind of something that we have to get done at some point in the near future.
Thanks for
Yes, and we so much appreciate that, Senate Bill 67. Illinois is not a RUSP alignment state, but they behave RUSP-like, and so we're very pleased that we're checking those boxes.
Thank you both for those updates. Do we have any questions? I think, Melissa, you put something in the chat. I'm sorry, I'm trying to catch up. If you
Yeah,
wanna...
you're multitasking.
Yes,
Dina, I was just curious. So how easy is it to predict stage of onset, either from the genotype or I guess from the screening assay itself?
Oh, that's a great question. So the first step in the flow is tandem mask, looking for sulfatides in blood to eliminate the false positives traditionally found with pseudodeficiency. That is then, as a second tier and third tier, includes an enzyme activity level, which is indicative of, so far, has been indicative. And also sequencing. There was a paper just recently published by a team from BioMarin, and Professor Gelb was involved with that as well, that indicates that they have about a 76% hit rate across, it's probably the wrong term, of the mutations that a baby may have. 76% of those will be indicative of the form of disease onset. What's not quite so clear in there, as I mentioned in my earlier comments, is that segmentation between early juvenile and late juvenile. And we're working to make sure that that data is distinct and divided, because historically we have not segmented those two forms of the disease. Over half the cases of MLD are late infantile, 20 to 30% are that collective juvenile population.
And in those cases, the late infantile, when is the ideal time to treat?
I can't speak for the company. What I have seen them do is treat at about 6 months of age. Typical symptoms would start showing clinically at 18 to 24 months, sometimes as early as 12 months. What they're doing is they want that baby to be healthy, have a little bit more body mass before they get the gene therapy. It does involve a transplant. The vectors that come in are corrected and so on, but underlying that is a transplant. So while we want to do that early, we want to do that when it's safest for the baby as well.
Thanks.
And Vineet, there's 2 questions in the chat for you. One is like, do you have any plan? Are you looking at your testing method? Do you have any comments on that? The question's asking, are you looking at a first tier method of C16 sulfatide and then a second tier genetic test? Go ahead.
Yeah, so again, I'm not the expert on that. I can't say for 100% certainty. I do know, yeah, the first tier is going to be I think it's a C16, as you mentioned. The second tier is an enzymatic confirmation, as far as I can remember. Yeah, I don't know. I'm not 100% sure. I'd have to talk to the scientist who's leading the validation for that, because that's not me. My expertise is more in molecular-based assays as opposed to biochemical. So I'm not 100% certain on that, but we are doing a first and second tier test for MLD. And then the second question, does Illinois have a required time frame to go live after the new disorder is approved through legislature? Again, I do not know 100% the answer to that question. I think there is a time frame that has been established. However, my knowledge and indication, as far as I'm aware of, is there is an understanding between like the legislature and our laboratory management as far as, hey, we need some time to go live with this. And the timeline that we did map out as far as Q2 of 2025 did seem to be okay by all parties, as far as I'm aware of. Again, I'm not involved in those discussions, so I'm not 100%. I can't give you 100% certainty on those details. I'm sorry. I'm not really much help here, but that's as much as I know of the situation.
Joe Orsini, Dr. Orsini, you may be able to comment on the C16. I'm also not a chemist.
Yeah, I was thinking that I'm pretty certain that they're going to set up probably the same model that Michael Gelb's proposed, which would be first tier sulfatide, second tier ARSA. Whether they'll do the genotyping there in Illinois or not, I'm not sure how that would work. But and if they would just go straight to a referral or not based on those two results, first and second tier. Probably they would.
Yeah,
But I'm purely speculating. That's what I would guess.
Yeah, that's again based on the meetings I've been in. It sounds like that's what has been proposed, and that's what the plan is. What I can do, I can talk to the lead scientist and have him join next month's meeting to kind of more give you kind of concrete details on what the plan is. Just so, again, this is kind of me explaining this secondhand, and I apologize about that in advance. I could have prepared if I had known this would have been a topic of conversation, but yeah.
No, I'm sorry, Vinny, that's my fault. I just saw the news and I thought I'd bring it up on the call.
Yeah, no worries. And if he's not able to attend, I'll make sure I get those concrete details and come back next month and explain those. Or I could email you, Jennifer, as well this afternoon or tomorrow and kind of confirm what those details are.
Yeah, absolutely. And we know that this is a new mandate. So, not all the plans are kind of planned out yet on how this is going to test. And
yeah, this news just came down like a month ago, like a couple of weeks ago. So we're all still kind of in the, oh my God, we need to do something about this phase.
Mm-hmm. Well, thank you both for your updates. This is very good information, informative, and will help the community. So thank you very much. I want to get through to North Carolina. We have someone on the call from North Carolina, Kate or Melissa or?
I'm here. This is Kate Kucera.
Hi, Kate. How are you?
Hi, everybody. Good, good. Wondering about your numbers. So we did finish DND screening in July, and I do have the final numbers. It's 16,563 babies. And so our screening is just one CKMM test, and then the rest of the testing is under a second consent. So based on the first CKMM, we had 95 cases, 58 males, 37 females. And then the second consent is for a NGS panel and for total CK testing at 1 month of age or later. So we had 80 consents for NGS and let's see, 68 for total CK testing. And we had 2 babies with DMD, both male, and 1 positive with CLCN1. So that's, I think you have all that. We didn't have any additional ones. So that's for the Duchenne pilot. So that was done under early check as a consented study. And then we have MPS2, that's a full scale separate study, so that's not under early check. We have screened over 130,000 and have one positive with an attenuated phenotype. And then I guess I'm going backward at it. So we had 22 screen positive cases, and only one of those was confirmed. That was the only one that had positive GAGs by the Fuller method at Revity. All the other ones were negative on dry blood spot GAGs. We are seeing some decreased activity of I2S on confirmatory as well. And some of them seem to have elevated dermatan sulfate. And so those are all pseudodeficiencies. Trying to see whether we have any left. I think we have a couple that are pending still for confirmatory.
And how long do you plan to will the pilot for MPS2 go on?
Yeah, so the original performance period was supposed to end at the end of September, but we are able to extend at least for a few months, and we're working on trying to extend further if we can make that happen. So that's in process.
Any other questions for Kate?
I suppose one other update would be, so we're also preparing a sequencing study, also in collaboration with GeneDx and Illumina. Our study also has two panels. One is highly actionable gene condition pairs that we evaluated individually. And then we have a second panel for less highly actionable. And then the third option for screening is genetic risk score for type 1 diabetes. And that will be done from the genome sequencing. So it's all genome sequencing backbone. GeneDx will be reporting the results from panel 1 and panel 2. And we are extracting the genotypes for the genetic risk scores at RTI to calculate and report that part. So the first panel, everybody will get the first panel, and the second panel and the genetic risk scores for T1D are optional. Yeah, so it's two monogenic panels, and the third option is the type 1 diabetes. I'm just looking what you're typing there. I guess we could call it a panel, although it has just one condition, the third one.
I will fix these notes in the once I read the recording, so I'm sorry.
No problem.
Do we have any questions? I know we heard a bit about the sequencing from a talk from Holly. I believe it was a couple months ago. I'm not sure if you guys got started, or at least it was early in the process. From last I remember, you guys were really early in this process. Have you guys started this the sequencing study yet?
So we have not started, but we're getting close. So some of the last things to put in place is the process, the pipeline for the type 1 diabetes genetic risk scores, because that requires data transfer from GeneDx and extraction of the variants and doing the calculations, and a kind of different pipeline that we are building on our end.
It's interesting, guys, that GeneDx has been mentioned both by New York and North Carolina. So it seems like as we're getting into sort of screening as diagnosis or screening informing diagnosis that GeneDx is, you know, starting to play a bigger role. So maybe something, Jennifer, to think about for future calls, including GeneDx.
Yeah, definitely.
Yeah, and also Illuminati as the platform, the current platform of choice.
Yeah,
That's a-
interesting. Thank you.
Mhm.
Any other questions for Kate? Okay, I'm going to switch gears again on you. It's a little bit of a different call, but I know that Nikki Armstrong for PPMD had some updates for us as well. So I was wondering if she's still on the call.
Yes, thanks, Jennifer. I'm here. Well, today is actually World Duchenne Awareness Day. So thanks for the opportunity to just update in where Duchenne newborn screening is. We are really excited about progress. As most of you are probably aware, at the August meeting of the ACH-DNC, the Duchenne package did move forward to evidence review. So that Duchenne package is under evidence review, and we're excited to watch that move forward. And we also had the first state pass legislation, yeah, you can see right up there, Ohio passed legislation to require Duchenne newborn screening. That did give I believe, a 9-month timeline for implementation. So we're looking at like springish of 2024 as I think the expectation there. But so lots of exciting things in Duchenne. I do want to just give a quick shout out and thanks both to the folks in New York State and the folks in NBS-TRN, and especially Amy Brower for all of their efforts in getting the Duchenne pilots, which was kind of the backbone to a lot of this work. Thanks.
Thanks, Nikki. We were glad to be part of it. And I'll never forget how many exons the gene has because of the September 7th. I use that when I teach a course about it. So it's such a great milestone.
Thanks, Amy.
Any questions for Nikki? Thanks, Nikki, for joining and giving us the updates. Anybody from the feel free to ignore me if you have to, but I thought I saw someone on the call from Ohio. Oops, I don't know what happened with my slides. If they wanted to comment about the process for screening for Duchenne in Ohio.
Yeah, this is Sharon.
Hey, Sharon.
Hi. We are in the process of trying to we've determined that we will be using GSPs for the PerkinElmer kit for testing for Duchenne. We're trying to get something up and running in 240 days, which is what the legislation says, is very tight. So we are trying to get another GSP, so we'll have enough machines to be able to do the testing. We are trying to figure out whether, a timeline for updating our data system to be able to report results. And we are trying to determine what the appropriate follow-up is when there really is no treatment available in the, infant phase. So we are really, let's say, struggling and working hard.
Thanks, Sharon. I'm not sure if we have any other questions for Sharon or Nikki. Oops. I will provide unfortunately, Angela was not able to, she had to jump off to another call as you guys are very busy. But she did send me some information about updates for some of the screening processes. She did say for Krabbe, they have sent 58 specimens for cytosine testing. There's only still one screen positive person. And then for MPS II, they have sent 87 specimens for GAGS testing. They have 7 children that were positive. 4 were pseudodeficiencies. 1 was normal. 1 refused further testing, and there's another result pending. I will not be able to answer any questions on their screening process, but feel free to email Angela if you have any questions about her update. And with that, I'm going to turn it over to Amy, if Amy wants to make the announcement for the our upcoming research summit that's coming this week.
Yeah, Jennifer, if you could just go to the previous slide, just to let everybody know, we've got a new podcast that's out. Effie Parks is a leader in the rare disease community. So she talks a little bit about her journey with her son with a rare disease and all her efforts with Global Genes and other activities. So if you want a lot of laughter and a little inspiration, feel free to check out that podcast. And secondly, next week, we're really excited to host the 2023 Research Summit. Just to remind you, our Research Summit is a little different than the network meeting where we talk about people who are using the MDS-TRN. The Research Summit is to sort of talk about the future and set the stage for future activities that may be involved in newborn screening research. So the next slide details our speakers, and we'll be putting out some social media so you can sort of look at the events, and Jennifer can put the link in the chat window to register for the meeting. So we're going to go beyond the newborn screening period and have some reports from efforts in the preconception and the prenatal time point, as well as hearing about some ways to interpret genetic and genomic variants using protein signatures, a little bit of artificial intelligence, and some new ideas focusing on some efforts that are funded by NCATS, looking at some rare diseases across the world. So we're really excited. We've got speakers from France, from Latin America, and from some other countries. So we hope that you'll be able to tune in or listen at your leisure once they're posted on our YouTube channel.
Thanks, Amy, for the updates. I'm going to go ahead and click on this slide, and well, oops, we will get you the link to the website. And please join us for the next meeting. Our meetings are always the first Thursday of each month. So look out for announcements for the Research Summit, as well as our next meeting. So with that, I can close it out, and thank you everyone for joining.